So we just want to start by welcoming everyone and thank you all for making the time in your day to listen, learn and grow with us as we dive into this conversation with Joel Williams around foliar nitrogen. We're living in a time where we're facing ever-increasing costs on farm due to inflation and labour costs and fertiliser costs and things like that. So we believe there are going to be some pieces of what Joel will share with us today that will shine light and encourage and perhaps even challenge us on our thinking towards nitrogen use, how we can drive more efficiency, how we can be better stewards of our limited resources, build soil biology and a healthy ecosystem that sustains our production. I'm Joel, I'm the marketing manager at Metalform. We're the designers and manufacturers of the tow and Fert. And with me today is Tim. He's a commercial manager here at Metalform. Tim, do you want to tell us a little bit about yourself?
Yeah, sure. Thanks, Joel. Yeah, I'm pretty excited to be part of today's session with some great minds sharing a bit of their work and I guess their passion on how it can integrate with tow and Fert users. I've been with Metalform for over 20 years now and have been a part of tow and Fert since its inception. So I've been along with the ride. And also it's continuing and ongoing development, which we've got a lot going on in that space, which you'll hear about later this year. I guess over these years, we've seen a lot change, and we're pretty stoked to be part of helping agriculture on a whole, helping achieve a more sustainable and financially viable system. Over my time with tow and Fert, we've seen it become a super useful tool around the world, where it all started in the dairy space. But more recently progressed into being used in sheep and beef, the tree crops, viticulture, vegetables, and just in the last few weeks in Australia and the broadacre cropping market. So yeah, it's an exciting space to be part of. So yeah, thanks Joel, Canaan and Joel, but that's enough from me and I'll hand it back to the experts.
Awesome. Thanks, Tim. Speaking of the experts, we've got helping us today is Canaan from Agrownomics. Canaan, would you be able to give us a bit of intro?
Yeah, hey all. Yeah, I'm the CEO of Agrownomics. We are a South Island-based consultancy firm around soil health. Have a bit of a background around dairy nutrition, and dairy is probably my sort of favourite lane, or close to my heart. But we have a team of 10 consultants, and we sort of range in viticulture, around sheep and beef, dairy and arable. So looking forward to today's discussion. It's going to be great.
Awesome. So before we introduce you to our guests today, we just want you to take a little mental breather, get off the farm and tune in and be present. It's not often we get to experience a world-class teacher, but we need to still the busyness in order to capture the gems that we're going to hear today. We have the privilege to welcome our guest, independent plant and soil health educator, Joel Williams. Joel has an extensive experience in working in Australia, the UK, Canada, particularly well known for his integrated approaches to sustainable food production. Welcome, Joel. We're really glad to have you here and we're excited to see what you're going to be sharing with us today. Obviously, you're going to go through a presentation followed by a bit of a Q&A where the listeners can chuck in their questions in the Q&A tab here in the windows they see. Before we begin that, can you quickly share your journey to this point and where did your passion for soil and plant health begin?
Thanks very much, Joel. Yeah, great to be here and looking forward to the session today as well. And yeah, for me, actually, the interest in soil started for me quite a long time. It was a good 20 years ago. I got right into growing. This was even when I was probably about a teenager. And I didn't actually come from a farming background, but by the time I finished high school, I was really into growing things, a lot of vegetables and things, and just knew that I wanted to go and study soil and agriculture. So yeah, I pretty much dived straight into it from there. And yeah, it's funny, 20 years ago, soils weren't quite as fashionable as they are these days. I used to get funny looks and raised eyebrows when I would tell people that I was at university and studying soil or specializing in soil. I used to get raised eyebrows. So these days, it's a very different story. It's great to see that that so many farmers all around the world are really engaging with this important topic of soils and soil health and that kind of thing. So I studied in Brisbane in Australia. That's where I'm from, as you mentioned. So I have quite a bit of experience there. But I also moved to the UK, and I lived over there for 10 years and worked quite a lot, particularly in the UK, but all around Europe as well. And then 6 years ago, almost 7 years ago, I moved here to Canada, where I've been based here since. So lucky enough these days to be traveling all over, particularly those three areas. I do lots of work mainly in Australia, Europe, and North America is where I'm most active. So doing primarily educational work, as we'll do today. So yeah, shall we dive into the presentation? I'll share my slides. I was around at my neighbor's house yesterday evening. He has this beautiful, springtime here, of course, and it's he has a beautiful tree that's just blooming, and I was around there having a look yesterday and having a good sniff, and I'm regretting it today. I've been so stuffy and congested today, so I've got a box of tissues with me here next to the computer, and I might need to give my nose the odd blow here or there, but hopefully I survive. I've just been really stuffy all day, so
I feel you there.
I'm regretting it now, but okay, so you can see my screen okay there?
Yep, that's looking good.
Awesome. Okay, so let's dive into it. Feel free, as Joel Crump there mentioned, throw your questions in, and we'll have into the chat box there, and we'll have a good chat at the end. So the topic today was foliar nitrogen. We'll extend that a little bit and just talk a bit some other aspects of plant nutrition, foliar feeding, efficiencies, some of the broader impulse in terms of sustainability and nitrogen management. So we'll kind of cover few topics and threads, and we'll weave them into this particular topic with quite a focus on where foliars can sit within our broader nutrient management. So, as I'm sure many of you will be aware, nitrogen is a very important nutrient, very important for production, but it is a double-edged sword. It's also a very reactive, a very leaky nutrient, and that makes it particularly problematic for its management. So it can off-gas into the air as ammonia or nitrous oxides, or of course it can leach through the soil into groundwaters, into rivers in the nitrate form, something I know is a big problem for you folks down in New Zealand as well. So it's a very challenging nutrient because, of course, it is so important and we do need it. It is one of the important macronutrients, but it is so reactive and so leaky. So it's very, very difficult to optimize the efficiencies of nitrogen when it has these tendencies to easily be lost from the system. And those losses that we're talking about are primarily, they're all functioning, or they're all happening through the inorganic pathway. When nitrogen is in the inorganic forms, that's where it's particularly vulnerable. So we'll talk about the importance of that as we move through, and how that differs in organic forms of nitrogen. They're a lot less leaky and a lot more stable, and also bring other benefits for plant health. So it's this problem of the leaky nitrogen, it is very much a global problem. There are a couple of hotspots, particularly in the world, North America, Western Europe, and many parts of Asia there that you can see. So we do have problems of over-application of nitrogen in relation to the production that is achieved. And that's actually what this, specifically this image is demonstrating, is illustrating, is the nitrogen use relative to the productive outputs. And you can see that we do have excess moving into the ecosystem. It is that leaky nutrient that then, once moves into the ecosystem, can cause a lot of problems. And that can be imbalances in the plant. And this point about nutrient imbalances, this applies whether we are talking about our productive plants, our crops, our pastures, or if we're talking about the native plants and the native systems in our ecosystems. But it is this excess of nitrogen within plants which then becomes imbalanced, and this can undermine plant health. and plant function. And so when we undermine plant health, the plants can suffer from that, of course. This can lead to things like loss of biodiversity, excess nitrogen in the soils. This has impacts on nitrogen carbon balances and all of the greenhouse gas and carbon fluxes and things that come with that. And then, of course, the nitrate form particularly is the culprit for the water contamination. So this leads to low nitrates in the waters, in our lakes and streams and things. This leads to low oxygen, algal blooms, acidification of the water. And of course, nitrates in the drinking water is a big problem for our livestock and, of course, for human health as well. So there are many, many good reasons why we do need to be careful with nitrogen. It does cause a lot of damage, a lot of problems when it escapes out of our fields, out of the soils, out of the productive areas where we need it. And you would all be very familiar with the fact that when we apply nitrogen onto legumes, legumes get lazy. They don't fix that atmospheric nitrogen when they have too much nitrogen in the soil and in the system. And indeed, this is also true for non-legume, non-rhizobium nitrogen-fixing bacteria, some of the other free-living and associative bacteria who can associate with grasses, with cereals, with all sorts of plant species, not just legumes. These organisms, these bacteria, are also in the same way that rhizobium and legumes get lazy with too much nitrogen. These other free-living bacteria are also suppressed by excess nitrogen. That's what this study here was looking at, 35 odd years of nitrogen fertilization, and they found that that N fixation was drastically reduced by around about 50% after those almost 4 decades of fertilization. This suggesting that that long-term fertilization has selected against that process of nitrogen fixation and against the specific groups of organisms who are responsible for that, these diazotrophs. So yes, nitrogen also is suppressive of the very process that, of course, we want to optimize and maximize. This is the free nitrogen from the atmosphere. There's plenty there, 78% nitrogen in the air, nitrogen gas. We want to be maximizing and optimizing the free supply of that nitrogen from the air, and the use of nitrogen fertilizers undermines that very action that we are trying to enhance and support and get as much free nitrogen as possible. And I mentioned there in that earlier slide about biodiversity loss, how when we undermine plant health, plants can then die out or become weakened, and then they are outcompeted. And this is a really nice example from a long-term study at Rothamsted, also on a grassland, where we're looking at the control versus the fertilizer. And we're looking at the plant species diversity here. And you can see where we have lower nitrogen, we have greater plant species diversity in those pastures. But as soon as nitrogen starts to become enriched in that soil system, we have, certain plants that then are nitrogen scavengers, like some of the grasses. So they begin to kind of dominate and outcompete some of those other minor species that may not be so happy under that nitrogen enrichment. So you can see here through this, we've lost some of the plant species diversity under the fertilizer treatment here as compared to the control. So that's a kind of a quick overview of some of the concerns and the reasons why we need to focus in on optimizing our nitrogen management. Those are some of the negatives. And so that leads us into this discussion of, well, how do we transition? How do we be much more judicious with our nitrogen inputs? How do we improve the efficiencies? How do we use less and get more? And I'm a big fan of this particular framework called ESR that kind of thinks about this, conceptualizes this concept of transition. How are we going to transition farming systems from their high input dependency towards something more sustainable, whatever we want to call that, but towards a system that is less dependent on those external inputs. We all saw this a couple of years ago with the nitrogen fertilizer price hike. All of us were impacted by this all around the world. So is any way in which we can minimize our dependency on nitrogen fertilizers is going to help with the resiliency of our farming systems. And I'm a bit of a fan of this kind of particular framework that helps to conceptualize this. The ESR stands for efficiency, substitution, redesign. And really all it really talks about is that it's just a way to think about that transition process, saying, okay, if we are currently very dependent on inputs, and it could be nitrogen in this example, but it could be other things, pesticides, or other aspects too that we could think about this. But the initial point is that we start off, we are dependent on those inputs. How do we start that process of transition? And the first one is efficiency, meaning we need to increase efficiencies. We need to be as efficient as we can with that input, meaning that we can start to use less. Because by improving efficiencies, less losses, less leakage, therefore we can improve those efficiencies and use less. So the efficiencies focus is kind of a really initial big one that says, okay, I'm still going to use the input, but I want to get better and better and better at using less and less and less. And you're going to kind of really focus on all of the things that can help improve that efficiency. And these are not mutually exclusive in terms of 1, 2, 3, in terms of their order. You can kind of be doing these things all in tandem, but I'll just kind of explain them in 1, 2, 3 format for the benefit of the concept. So after we optimize the efficiency, we can think about substitution. And substitution then says, okay, is there an alternative? Can I use something else instead of that input? Can I substitute in with an alternative that is maybe a little softer, not as damaging to the environment, a little less leaky, something you're still using inputs, but maybe you're using a different suite of inputs, more of the organic forms of nitrogen, for example, organic amendments, compost, these types of things as an alternative. So you're thinking about substituting into softer options, better options, normally more biological type based options. And then last comes redesign, and that's where you're really saying, okay, how do I move away from product dependency completely, not just synthetic inputs, not just alternative bio type inputs? How do I move away from some of these completely in a redesign, meaning I redesign the farming system so that I don't need any of these inputs? And that, in this example, redesign really very much centers around the role of legumes and bringing more plants diversity in, particularly legumes, who will perform that function of nitrogen fixation so that we still have nitrogen in the system for our productive goals. So that's kind of the idea of the ESR in a nutshell. And so just as an example, in the case study with nitrogen, if under the efficiency category, I would put foliar nitrogen there. We're still using synthetic nitrogen, we're still using these forms, but we're going to be really targeted in the placement of that through this liquid, through the foliar application onto the leaf, and thereby minimizing losses, using less, improving efficiencies. So foliar nitrogen would be a good example that sits under that category. Then you've got other things like the inhibitors, they help to slow the losses, of course. Other cultural practices like maybe split applications, spoon feeding, low dose, more frequent spoon feeding. That's going to help to synchronize supply and plant demand. So again, therefore minimizing losses. And then other carbon-based kind of stabilizers, carbon-based inputs. I'll talk about those briefly shortly. So a substitution strategies would be, okay, can I use nitrogen fixing bacteria? Are there some other bacterial products out there that we could use in place, or some of these organic forms of nitrogen, amino acids, which are also very favorable, more stable, less reactive. So we're still using those inputs, but we're substituting to alternatives that are a little less leaky, a little more sustainable. And then there's that redesign. So really it centers, as I mentioned, around diversification with legumes, maybe better rotations, maybe livestock integration, better grazing management. So really we're talking about parts of the system, how we manage the farming system, and how we can tweak and fine tune the farming system itself in order to optimize these processes. So that's that kind of idea of ESR. And ESR, efficiency and substitution, they're both really important stepping stones. They're really important pieces of the puzzle. They start that transition, they get that process moving, and that helps us to start using less of, for example, that synthetic nitrogen, which is going to have suppressive effects on the nitrogen fixation that we talked about earlier. So we are beginning that process of using less so that we can start to increase or optimize some of those redesign elements, like harnessing and fixation as an example. So yes, efficiency and substitution, look, they're great. They're really important first steps, but there's a certain, they can only kind of go so far. If you really want to push towards a system that's really truly much more sustainable, there is much more greater potential in the redesign elements to make the farm system more self-sufficient, self-cycling, more resilient through that sense. So there's a lot of great potential in the redesign, but we don't want to get stuck in efficiency and substitution, but they are still very important first steps. And I'm a big fan of getting started on that journey and one step leading to the other, stepping stone kind of approach. So the redesign concept really centers a lot about diversity. How do we increase the diversity of our farming systems using more biodiversity? And typically that's more plants, but it could also be livestock integration, would be a good example of that too, for those maybe cropping systems that don't have livestock. But for already a livestock-based system, it would center around how can we use other plants to increase the diversity, and that's going to bring some of these redesign elements. So, there's a long list of kind of examples here. I won't go through them all, but it could be things like simply rider rotations, other cash crops, or using just different plants, annuals, perennials, summer, cool season, warm season types of things, covers, companions, intercrops. Okay, in a grazing context, okay, we're going to be focusing more on multi-species pastures. There may be some scope for bringing in trees to that, some kind of silver pasture type arrangements, trees, pastures, and animals. I think there's a lot of really interesting things happening in that space at the moment. But it could also be other non-productive plants, things like our biodiversity strips or shelter belts, your field margins, buffer zones around our waterways, those types of things. So all of these different uses of plants increase the diversity, and that really helps to in this redesign element, because what redesigning the farming system with plant species diversity does is help to make the farming system a lot more multifunctional. And this word multifunctional is really something that our farming systems are needing to move towards. We've traditionally been very focused on being very productive. And look, that was the good thing in post-World War II, food shortages. Yes, it was important to increase productivity and to feed the world. This was the right policy action at that time. But of course, it's come at some hidden costs of that we're becoming more aware of the environmental decline. We've been chasing productivity, not putting that productivity in the context of the local landscape. How do we optimize production within the context of the health of the ecosystem and the landscapes in which our farms sit? So yes, productivity has been good, but we've not been kind of as optimizing some of these other ecosystem services. A natural ecosystem is the opposite. Yeah, they're very good at providing ecosystem services, but they're not so productive. So how do we strike this middle ground of supporting the ecology and the landscapes in which our farms sit, but also still being productive? And we need to kind of be thinking about how we can find a middle ground between these two. And that's where plant species diversity adds multifunctionality. And so this really helps with the sustainability of our farming systems. We're still going to be focused on production. We have to be. We've got to pay the bills, of course, but it's about how do we do this in an economic way and in a way that can also support the local and surrounding environment and waterways, is a good example for you folks in NZ. So I won't dwell on this. That's just a visual image to kind of emphasize this point. It's really about using a diverse array of plants, particularly easy in a pasture context. Okay, in a cropping context, it's much harder to be highly diverse. Maybe just a companion crop or an intercrop is as far as you can go, maybe up to 2 or maybe 3 plant species. But in a pasture, you've got a lot of opportunities for however way mix, 8-way mix, 10-way mix, 20-way mix, whatever. We can have those debates about how many should there be. There's no magic answer to that. It really depends on the context. And I think there's a lot of value that comes even in some of the simple 6-odd way mixes. I think there's a lot of good value in some of those. There's a lot of heavy lifting in the initial jump up from increase in plant species diversity, I think. So I think it's really easier in a pasture context, and that's where I'm encouraged to see all of the discussion about multi-species pastures, multi-species swards that's happening all around Europe, in Australia, here in North America, everywhere. We're seeing a lot more engagement in a bit more diverse pastures. So today we're going to talk about foliar nitrogen as a really important piece and a strategy of an integrated nitrogen management approach. And that really just means we're going to be integrating many tools in the toolbox to overall manage this important nutrient, to manage it for optimized production, manage it for optimized efficiency and economics, and manage it for minimal leakage, minimal ecosystem decline. And so silver foliar sprays is a fantastic tool. I'm a big keen promoter and a big fan of it. But you know, it's not a silver bullet. It's not the be all and end all. It is one very valuable tool. When integrated amongst other tools, can really make a big impact and make a difference and be a piece of the puzzle, be a part of the system. So yes, we're going to go now and focus deeper into the specifics on foliar nitrogen, but just to emphasize that there are other things that we should be doing and that we can also integrate as part of our overall strategies: carbon stabilizers, organic amendments, some of those biofertilizers, and fixing bacteria type things, plant breeding, so breeding varieties with bigger roots that will capture some of that nitrogen before it leaches away, those inhibitors that we talked about, precision, variable rate, some of these things, cover crops, catch crops, green manures, you know, preventing losses or bringing some free nitrogen into the system, companions, intercrops, multi-species pastures, livestock integration, you know, better grazing management, all of that kind of thing. So just to emphasize, look, there's many other things that we should also be thinking about too. So efficiencies of foliar sprays, are they really more efficient? And look, there's a whole array of papers out there. I'm going to share a couple of examples that suggest, yeah, look, foliars can be efficient and more efficient than soil applied. But you know, it depends, and it depends on a lot of different contexts, and we're not going to go deep into all of that discussion today. But I just want to emphasize, look, there are examples of why foliars can be and very often are more efficient. And some of those are really simple, and some of them are a bit more nuanced. And so, for example, okay, even coverage compared to sporadic placement of granules. You know, you get a liquid solution of that nitrogen, perfectly liquid, even coverage coating over the foliage. We get a very much more even application compared to sporadic placement of granules where they solubilize, and it's a bit more uneven in that sense. With the foliar application, there's also less of these nutrient lockups and antagonisms. You know, nutrients in the soil, they get locked up. They interact with other nutrients and they can become unavailable. We can bypass all of these issues if that liquid is going straight onto the foliage, into the foliage of the plant. We minimize some of those lockup-based antagonisms in the soil. Of course, we have less of these losses that I've talked about already, less of the ammonia volatilization, nitrous oxide, denitrification, there's off-gassing, less nitrate leaching from a foliar spray. Foliars can also help to maintain product supply of nutrients in suboptimal conditions. So for example, under drought, when the soils are drying out, it's harder for the plants to get nutrients out of the soil. So the foliars can help to maintain nutrient supply during those conditions where root activity may be impaired. And then we also have an opportunity to use very specific forms of nitrogen, which have an efficiency gain. Different forms are more and less efficient. And I'll talk about this at the end of the presentation, just to say that there are efficiency advantages to some of the organic forms, like amino acids. They are very beneficial forms of nitrogen for the plant to use. They save the plant a lot of energy. So we'll talk about that briefly at the end too. Now, part of the efficiency gain is also just really simple, that you're using less. And when we use less input, we are operating more at this lower point of the classic kind of nutrient efficiency graph here. This is, you would all be aware of the plateau, of course. As you move up towards the plateau, it becomes the yield, the law of diminishing returns. You know, the yield gain per unit of extra application that you're putting out gets smaller and smaller and smaller. So when you're operating up here, yes, that's just a natural part of that curve. When you lower the rate and use less fertilizers, well, we're bringing the efficiency down into this much steeper curve that you can see here. So for each extra kilo of nitrogen, we're getting much bigger benefits, bigger return on investment. So part of it is, there are some special things about foliars, but part of it is also just a law of when you start reducing the rates, you will get more efficient gains per unit of nitrogen when you begin to operate down at this part. And that's kind of where we want to optimize and not get too high up towards that plateau. But beyond that, the foliars actually can also enhance the uptake of nutrients from the soil. So foliars can enhance the uptake of the reserves that are already in your soil. And we have various examples of this. Here's just a couple, for example, where a foliar ammonium application increased nitrogen uptake from the soil, foliar potassium actually increasing potassium uptake from the soil, or foliar urea enhancing both nitrogen and potassium uptake. And the mechanism by which a foliar can stimulate soil nutrient uptake is because some of those foliar applied nutrients, they can be translocated down to the roots where they can stimulate root production, root biomass production. So we're growing a bigger root system, particularly when foliar nutrients are combined with some of the biostimulants, which can also stimulate root growth. So now we're just simply using foliars to grow bigger roots to therefore acquire more nutrients from the soil volume, from a greater soil volume, and therefore just improve the uptake and utilization of what's already there in the soil. So that's kind of one mechanism, but we also just have increased root exudation. When we foliar apply nutrients, we prime photosynthesis, prime sugar production. Some of those sugars are sent to the roots, exuded out of the roots to feed the soil biology. And then, of course, that soil biology is also going to cycle nutrients from the soil and deliver those to the plants. So through these two mechanisms, foliars can actually improve the utilization of soil nutrients. And that could also include some of the soil fertilizers, soil applied nutrients, or the nutrients that are already present in those soils. So here's two quick examples of looking at the efficiency discussion of foliars versus soil. Both of these are from New Zealand in a pasture context. Here we're looking at urea in the granular liquid, and then also with the urease inhibitor. And you can see here that where they're looking at the efficiencies here. So where they applied the granule, they got 10 kilograms of dry matter production per kilo of nitrogen applied. So that's from your standard granular urea. But where they did a liquid urea, that went up to 19, a foliar, so that went up to 19 kilos dry matter production per unit of nitrogen. And then when they added the urease inhibitor on top of that, in that liquid, it went up to 23 kilos production per unit of nitrogen. So we've gone from 10 as a soil applied granule to 19 as a foliar, and up to 23 with the foliar plus urease inhibitor. So here's a nice example of, yes, per unit of nitrogen, the foliar increased the production and therefore was the most efficient per unit of nitrogen. And this is a busy slide. I apologize. I'll just drag your eye straight down here to this part of the slide here. This is kind of a nice summary slide that's also looking at the efficiencies. And sorry, I know it's a bit small. Hopefully, I'll quickly talk you through it. We're just comparing here a couple of different sources of nitrogen in a granular form with also a kind of a liquid type application. So you can see here we have, looking at the efficiencies, urea granule, urea G, you can see, and there's our, again, biomass production per unit of nitrogen, whereas we do the urea in a liquid, and you can see that we have an increase in efficiency there. Same with urea when combined with the agrotane. Here's our granule, and here's our liquid. So we see this nice little increase here with the liquid form. Oops. Here we have ammonium nitrate granular versus ammonium nitrate liquid. And again, we see a nice bump here, and ammonium sulfate here, granule versus liquid. And you can see, again, consistently, the liquids are increasing production per unit of, we are increasing production per optimizing that efficiency per unit of nitrogen applied. So yes, here's a couple of examples in summary whereby through that foliar pathway, of course, we're minimizing some of those soil-based losses, and therefore per unit of nitrogen, we are able to increase production. So why foliar feed then? It can increase those nutrient use efficiencies, and that will improve the economics. You're going to need to spend less money on fertilizer. It's an obvious win-win, and it's also a win-win for the environment because we're using less escaping out into the ecosystem. So we can also use foliars to improve yield, quality, metabolism of plants. We can reduce those negative effects on the environment and also potentially increase feed value or food value for health benefits, be that for humans or for livestock. Now, of course, the problem with soil uptake, look, roots were designed to take nutrients up from the soil. I'm not anti-soil applied nutrients and soil-based strategies. They're also part of the system. This is not necessarily having to say that a foliar can supersede a soil, although there are some interesting examples of that happening. All I'm just pointing out is that, yes, soils have their place and so does foliars. And we just want to try and optimize and maximize both of those. They have strengths and weaknesses on both sides. And we just want to optimize both of those processes, again, in an integrated approach. And the challenge with soils is that, yes, roots are well adapted for nutrient uptake, but soils are highly dynamic and tricky environments where nutrients can be easily locked up and lost or cycled, etc. And so with the example of nitrogen, as we've talked about already, it can leach, it can off gas, it can erode, run off in the surface through erosion, run off in the soil sediments. So nutrients can be lost from the soils, but even if they're not lost, they can also just be locked up or antagonized. So they can bind to soil particles and be not taken up. They can be locking up with other minerals, whereby preventing the plant from taking them up. So again, just because a nutrient is in the soil doesn't mean it's actually available and being taken up. So there are these antagonisms, these interactions and these losses from soils, in which can make them very difficult environments also for taking up nutrients. And that's where foliars can bypass some of those issues and again be a really good tool in the toolbox. And so here's some of those nutrient use efficiencies of soil applied nutrients. And you can see just some various examples here. These are kind of guideline numbers. You know, it's going to be different in different soil types, different environmental contexts, different climates, etc. It's going to be variable, but these just give you a guideline here. You can see that, you know, nitrogen is particularly a worry because we use so much of it, and it often hovers around about 50-odd% use efficiencies. About 50-odd% of what we apply is taken up by the plants. That other 50% potentially lost. I mean, some is held in the soil, of course, in the soil organic matter, but some is also potentially lost. And you can see other nutrients are also very low efficiency when they're applied in the soil. Phosphorus, not so easily lost from the soil, but easily locked up, as I'm sure many of you would be aware, particularly under acid soils and that kind of thing as well. So very low efficiency, fertilizer efficiency of phosphorus, 10, 15, 20-odd% of what we apply is taken up. The rest is often locked up. And you can see as we go down that list, particularly some of the trace minerals, the efficiency really drops right off. And that's really just because trace elements are, of course, required in such small quantities. And when we put such small quantities of nutrients into that big soil volume, there's a lot of soil volume there, which can be rapidly lock up those nutrients before the plants have the ability to take up those nutrients. So that's why foliars, even amongst people who are more skeptical about foliars, I think many of them would even still agree that foliar applications of trace elements is very relevant and effective because they do have this high tendency to lock up, and they are required in small enough amounts that a foliar spray can supply meaningful amounts of those nutrients. So that's what we're working with in soil-based efficiencies, and that's what we want to try and improve by bypassing that and delivering those nutrients straight into the foliage. So do plants take up nutrients through the foliage? I mean, is it a natural thing? Does it happen in nature? Is it normal for plants to be taking up nutrients? Why do they have roots if they can use these foliar nutrients? Well, again, it's just about optimizing both pathways. Both have pros and cons. And yes, there are examples in nature where plants do take up nutrients through the foliage. Seaweed, of course, kelp would be a very good famous example of that, growing in the mineral-rich ocean water. This mineral density is what drives incredible photosynthetic rates, incredible biomass production that we see with kelp and seaweeds. So they'd be a good example. Even dust storms, believe it or not, have been shown to supply meaningful amounts of nutrients through the foliage. And particular example here is looking at wheat and chickpeas, where they applied dust to the leaves, and they showed that particularly those two plants were very effective. They actually acidified, they produced leaf exudates and organic acids, and they acidified the leaf surface, and they actually solubilized meaningful amounts of phosphorus out of that dust, and whereby they used it for biomass production. So even dust deposited on the leaf can be then acidified and solubilized. Nutrients can be solubilized out of that dust and taken up through the leaf. Another example would be insects and waste material, or even us, urine. Plants can take up urine through the leaves. They can actually take up insect feces. They can absorb amino acids, organic forms of nitrogen from insect feces. So yes, plants can take those up. All of our various animal waste products can be taken up through the leaf, and we have good examples of this in the literature. And lastly, even fog. This is from a great study on Californian redwoods, that's still demonstrating that even from fog, plants will take up ammonia gas. That gaseous ammonia form that we were talking about, we're trying to minimize some of those losses. Actually, that can be breathed in through the stomata and utilized by the plant as well. So yes, it is a natural thing for plants to take up nutrients through the foliage. And yes, it can therefore be a tool that we can work to optimize. So I won't dwell on some of these uptake pathways. I want to kind of get into some of the practical nitty-gritty on how we kind of design a foliar spray based on kind of this. But just to say, look, there are well-known documented pathways in which nutrients are taken up through the leaf. The main ones would be those top three through the cuticle. That's that waxy layer on the leaf. There are these kind of micro pores, micro holes in there, which kind of expand and contract depending on things like humidity. And when those pores are open, nutrients can pass through that waxy layer, through these micro pores. So that's the main one. Then we also have, of course, the stomata. When the stomata are open for breathing in carbon dioxide, we can also foliar apply nutrients at this time, and nutrients can pass through the stomata particularly when they're open. And even trichomes, and this is a newer one. Trichomes are some of the fine hairs on plants, on plant leaves. And it turns out that some of these trichomes are very effective at capturing things like water droplets from our foliar sprays, or rainwater, or even dust particles, capturing those and funneling them down to the base of those trichome hairs, where their nutrients can then be absorbed. So those would be the main ones, but there's also a couple of others, things like veins in the leaf, other epidermal structures in which, like things like bark on trees, for example, has also been shown to be absorbed. So there's a few other mechanisms by which we have documented uptake. And here's just a nice image of the kind of top one there is the cuticle, and this is what I was talking about. Here we have, under high humid conditions, those micropores in that cuticle are more open, and therefore that solution, the nutrients in that solution can pass through, as compared to low humidity, dry air, where those micropores are more closed and retracted, and therefore we're not going to get good uptake of that solution because the pores are very closed. And of course, similarly with the stomata, there's the guard cells of the stomata. When they are nice and open, again, high humidity, those nutrients can pass along those guard cells through the stomata and onwards into the plant. So that's those two examples. And this was the one I was mentioning there about the trichomes. Just a nice little example. You can see here's the fine hair, and here we have a droplet that has been caught by that hair and kind of funneled down to the base of that hair. And it's quite interesting. What they showed in the study here was that these cells right here, these are called the basal cells, meaning the cells at the base of the trichome. And it's specifically these cells that go around that trichome. They are incredibly permeable, even hugely compared to even just one cell away. Just this one cell next door, the permeability drops right off. It's a very specific function to these basal cells underneath the trichome that have very high permeability. And they were evolved that way, designed that way, to when the hair captures the dust or the droplet and it funnels it down, those cells are going to be highly permeable, ready to acquire the nutrients from that nutrient droplet, for example. So this is another emerging example that we are now beginning to understand more about. So how do we kind of design a spray for success? How do we avoid the spray and pray approach? How do we optimize some of the science about uptake and optimizing uptake? How do we use that knowledge to design a optimized foliar spray? And I'll break it down into these four categories here. The plant response ultimately depends on the formulation, meaning what's in the spray tank, what is the mix that we apply, how do we apply it, your nozzles, your settings, your speed, some of those other considerations. what is the crop or the plant species? Different plants respond differently, of course. And what are some of those environmental conditions? And I'll have to just summarize these. I'm mindful of the time. I'm going to kind of quickly flash up some slides and we'll talk about some of the key things here. But I'm also, there's a lot to this. You'll find some other talks on YouTube if you'd like to about this. I'll mention at the end, I did run an online course on foliar nitrogen earlier last year, and I go much deeper into this. So if anyone's interested, I'll flash up a slide on that at the end. But I'm going to cover some of the key highlights for now. But I just want to emphasize there is devil in the details. There's a lot of kind of points to kind of think about here. But let's cover some of the key ones. Formulation. So here's what goes in the spray tank. And I would just emphasize to start with that water quality, I think, is a really big one that is often overlooked by many farmers. You do want the water to be as clean as possible. And if you have especially very alkaline waters or waters that are high in carbonates or bicarbonates, these types of things, salts can also, high in iron would be another one. These things are contaminants that can bind to your nutrients that you solubilize in that water to foliar apply. So the more of those contaminants that are in there, the more they are locking up with your foliar nutrients before you've even got it out, before you've even applied. So I would emphasize, if you can get rainwater collection, invest in some rainwater harvesting and collection and storage, it's worth it. As clean water as possible is better. Even if you can invest in reverse osmosis, there are more farmers really going down this path and investing in reverse osmosis. And particularly on the cropping context, I know many farmers who are, for example, using herbicides, they're finding that when they invest in the RO, the reverse osmosis setup, that they see significant improvements in their pesticide use and pesticide efficiency. So particularly they're improving efficiencies of particular herbicides, reducing the herbicide usage because they're getting better kills and saving money. And so there are farmers I know who are investing in a reverse osmosis setup and paying it off within 1 to 2 years just on herbicide savings alone. So it does impact pesticide efficacy. It does impact foliar nutrient efficacy. So water quality is a really important aspect here. And I'd really emphasize people to look deeper into this one, use as clean water as possible. Now, of course, you've got other factors in the spray mix. How soluble is the nutrient? This is generally the more soluble, the faster the uptake. The smaller the molecular size of the molecule, the faster the uptake. I'll share some quick examples of this. Electrical charge, so cations versus anions, positively charged nutrients versus negatively charged nutrients. Both of those actually have some difficulties passing through the leaf because the leaf surface is negatively charged. So when you apply a positively charged cation to a negatively charged surface, there's a bit of a strong attraction there and there's a bit of a bottleneck, and it can slow uptake. Similarly, if you apply an anion, a negatively charged nutrient, to a negatively charged leaf surface, of course, there's more of a repulsion away. So this also minimizes uptake efficacy. So we want neutral charge, and that's where things like carbon additives and chelation is very important to neutralize the charge of some of those cations or anions. So some of those chelating agents are very important to include. Spray pH is also an important one. Most of your foliar nutrients are better absorbed under more acidic conditions. So I would say a maximum of 6, I think. I know many folks go, I would say even down to 5, 5.5 is also really good. I know folks that go even lower to 4. There's a kind of a whole range of kind of pHs that people seem to play around with. But I would say that I think a maximum of 6. And I think this is very important in the context of foliar urea because that urease enzyme that drives the off-gassing of urea, that conversion into ammonia gas, that enzyme, that urease enzyme, does not function under acidic conditions. So you definitely want the spray mix to be below 7, down around at least that 6 mark, where that urease enzyme will not be functioning. And therefore, we can also minimize some of those ammonia off-gassing off the leaf surface, which happens very minimally anyway as compared to soil, much minimally. But if we just slightly acidify the spray mix there, that ultimately helps to minimize that as well, but also improve uptake of some of the other nutrients. So, and then you've got other things like, you know, your surface tension. So wetter stickers, oils, some of those things can also really help. And I'll talk quickly about some of the carbon and chelation type inputs as well. I won't dwell on this one. This is standard stuff you all know. It's about your nozzles, droplet size, your pressure, your forward speed, all those types of things. The key message here is just make sure you get good coverage over the leaf. You want to maximize the coverage, the contact. You want to hit that canopy. You want to kind of make it stick. You want to make sure you cover that canopy. The more you can cover that canopy, the better the uptake. Now, when we're talking about foliar nutrients as compared to pesticides, of course, pesticides are very different. You've got to have, you know, finer nozzles, very, very even and perfect coverage to try and reach those pests. Whereas nutrients are different, you know, they are a systemic response. So even if you do have a bit of a bigger droplet that just lands on the leaf and some of the rest of the leaf doesn't get covered, that bigger droplet, of course, is going to be absorbed. It's a systemic effect. It will be absorbed through the leaf where it can be translocated throughout that leaf. So that's where, you know, even though the tow and fert uses much coarser, larger droplet, droplet sizes, that's okay as long as you still get that on the leaf and to stick to the leaf so that then that droplet has time to absorb the nutrients where it can then be translocated. And that's maybe a bit different from a pesticide application where you do need finer nozzles, but with nutrients, it's a little less critical. Then you've got the plant response in terms of the crop, FAC, or the species here. And this is really just saying that, look, different plants have different leaf structures, different canopies, different leaf area index. Some more waxy leaves, some less so. You know, brassicas are very waxy, of course. So different plants respond a bit differently to foliars in that sense because of some of these different leaf characteristics. And I think that, you know, let's use that example of the brassica and the waxy leaf. Sure, it has a tendency for droplets to really bead on those waxy leaves. So you definitely want to get a sticker there and reduce that spreader, reduce that surface tension to kind of spread out that droplet and get that to kind of not bead so much, but to kind of spreader stickers and get kind of good coverage on the brassica, because just to help to overcome those waxy leaves. But, you know, equally, some of the brassicas, they're a nice big broadleaf. They can have sometimes quite nice big round leaves. They've got a nice big surface area to capture a foliar spray as compared to, say, a grass, you know, a monocot. So there's trade-offs, you know, there's pros and cons kind of to everything here. So generally, younger leaves are a bit more responsive than older leaves. So that kind of fresh growth and fresh kind of flush of growth, these younger leaves, they're very, very permeable as compared to some of the older leaves that start to harden off a little bit. And don't get me wrong, those older leaves can still take up nutrients, but it's just the younger leaves are a bit better, a bit more permeable, a bit more sensitive to that. Okay, then we have the environment, and this is the other big one, the environmental conditions which you're applying. And the big one here is humidity. You really want humidity to be high because that's when the stomata are going to be open. That's when those micropores in the cuticle are going to be open. And so the magic number here is around about 70%. If humidity is above 70%, this is ideal. This is optimum for uptake. Now, don't get me wrong, you can foliar apply when humidity is a little bit lower than that, but you just want to make sure you've got things like some of those oils and things in there to kind of protect those droplets from the drying air, because what we don't want is hot, dry air that's going to dry out that droplet too quickly. We want a bit more of a slow and steady drying out of that droplet, because if the droplet dries out too quickly, we haven't had enough time for the nutrients to pass, to be absorbed into the leaf. So the nutrients can precipitate out, crystallize out, you know, onto the leaf surface, for example. So I'm just saying that 70% is optimum. So if you're pretty much 70 or above, you're in perfect conditions, and this is definitely worth aiming for. And therefore, you know, generally farmers would apply foliar sprays in the earlier mornings, in the late afternoon, into the evenings, because these are the times of the day when the humidity is up, and therefore the plants are more porous and they will take in the nutrients more effectively. Humidity and temperature tightly coupled, of course. Really, I would say that the lower the temperature, the better also. Absolute max, I've read a lot into this, and a lot of the literature says max 28 degrees. I think that's also pretty high. I would say, I think even if it was above 25, I think that's for me kind of a bit more my cutoff. And look, I know there's a practical reality here. Sometimes you've got to just go when you've got the window. And I know it may not always be perfect, but I'm just kind of sharing with you, well, these are the optimum conditions. So if you can kind of keep those temperatures, get in earlier or later in the evening when the temperatures are a bit cooler, the humidity is a bit higher, this is going to be better. A little bit of wind is good, of course. That helps with a bit of coverage. Not too much, of course, but a couple of Ks there is good just to kind of get that nice swirl of the spray and get it to kind of coat all over that leaf surface. And even drought conditions or drying conditions, foliars, as I mentioned earlier, can be very effective to still supply nutrients under that drying soil conditions. And I would just emphasize, however, that where they're most effective, where foliars are most effective, is more so in those earlier stages where you can really keep the plant holding on in drying conditions. Once the plant is very drought stressed, at the late stages of drought stress, I mean, it's really shutting down. It's not going to be performing well in terms of taking up a foliar spray. So foliars can really help in the earlier stages of dry stress, where they can really help the plants hold on through that dry stress much longer than otherwise. That's where I think they really have the maximum benefit. Okay, I can see I'm pretty much on the hour. I'm going to, if it's okay, team, I'll just carry on. I'm getting close to the end. I'll just take maybe another 5 minutes or so, just to quickly wrap up a few points here. I'll try not to eat too much into our question time. Okay, so I want to just come back to a little bit on this deeper, quick few points on the formulation, and just to emphasize the importance of adding carbon-based inputs with the nitrogen. And these are, as you can see visually on the screen here, these carbon-based inputs, they're just like a sponge, and they're going to bind to the nutrients. And this is very beneficial to help, particularly with the example of nitrogen, which is very reactive and easily lost from the system. It helps to stabilize that nitrogen, bind to, and minimizes that off-gassing, or minimizes the leaching if it was down in the soil, for example. So these carbon molecules, they bind and they change the behavior of that nutrient. That's very good to stabilize, minimize losses. But the other thing that this does is also improves uptake through the leaf. And this is that point I was talking about, whereby the cations or anions are not so effective to be taken up through the leaf. Whereas if we bind them with carbon, we've now neutralized that cation or that anion. We've made a charged neutral molecule that can then be taken up through the leaf. So the carbon-based inputs not only help to stabilize, minimize losses, but also help to improve uptake. And so I think they're very valuable. And that's why I'm sure many of you have heard of chelated nutrients and come across this kind of term. That's really what we're trying to achieve. We're trying to take a nutrient, we're going to wrap it up, and in this example we've got some amino chelated, binding some amino acids to that nutrient, whereby now we have neutralized the positive charge of that cation, for example. So now we have a neutral molecule, and now this can pass through the leaf much more faster, much more effectively, compared to the positive or negative charge, which is a bit more clumsy. It's not to say that it can't pass through, it's just not optimized. It's not going to be as quick, it's a bit more clumsy. So better absorption, better translocation. So the plants will amine themselves, even if we apply inorganic salts. The plants will immediately chelate them, often amino chelate them internally so that they can be translocated around the plant. So you've got a form that's kind of ready to go. And this can also help to lower some of that salt stress or the saltiness of some of those inputs. So, I think amino chelates are very good. I'll share some other carbon-based examples, but I'm a big fan of things like some of the amino acids and fish hydrolysates, just as an example. These are really good additives to add to the foliar spray to bind to those inorganic forms of nitrogen or other nutrients and chelate, complex them, and improve uptake. And here we have examples of how quick the uptake through foliar is. And this is one of its big advantages as compared to soil applied. So on the left-hand side, we're looking at urea. These are both on turf grasses. But here we're looking at urea on two different grass species here. And we're looking across time and absorption. Look how quickly it happens. It's literally in that first hour or two where a huge amount of that uptake happens. And pretty much, within that first 4-6 odd hours, most of the work is done. You'll still have some, dribbling along thereafter, but with particularly with urea, it's very quick to be absorbed. And so that's part of its gain, is that you put that same urea in the soil, you got to wait for that granule to solubilize, you got to wait for that to move down into the soil, to wash down, to be taken up through the roots, then translocated up to the shoots. So here we're illustrating an example that within 1 or 2 hours, we can already have that urea or that nitrogen in the plant and providing function. And over here on the right-hand side, here we're looking at amino acids. So these are some of those organic forms, carbon-based nitrogen. And you can see, again, we're looking at time. You can see how rapid the uptake is here. It literally happens within that first hour or so. So this is the big advantage of the foliars. It's the speed of uptake. The faster you get those nutrients into the plant, the less time they're going to be sitting around in the environment where they can be lost. The nitrogen is not going to off gas once it's in the plant. It's not going to leach once it's in the plant. So that's part of that gain, is that speed of uptake. And so here's some of the carbon sources generally that I'm kind of referring to. I'm sure many of you would be maybe familiar with these or using some of these. It could be as simple as a bit of molasses or some sugar. Humic or fulvic acids are very good. They're a very big sponge. They have a high exchange capacity to bind to a lot of nutrients. Amino acids, as I mentioned, I'm particularly a fan of. Great chelators, but also organic nitrogen, protein hydrolysates, seaweed, kelp extracts, other plant extracts, even using things like compost extracts for the humic type substances that are also in them as well can be used. So that's your kind of examples of all sorts of different kind of carbon-based additives that ultimately help. And the urea is, of course, a very special form of nitrogen because it is already carbon-based. It has carbon in the molecule and the structure. Now, you put that urea into the soil, however, and that urea doesn't stay as urea. It will convert back. You will lose that carbon as CO2. Water will mix in there, H2O, and you will lose CO2, and that nitrogen will be turned into ammonia. And then from there, it can be turned into nitrates, of course, depending on your soil conditions. So when you apply urea to the soil, urea does not stay as urea. It converts back into ammonium and or nitrate. But if you apply urea onto the foliar, now you are getting nitrogen into the plant as urea, as carbon-based nitrogen. And there's an efficiency gain that comes with that carbon. And that's why urea is so fast to be absorbed, because it is not a cation, it is not an anion, it is a charged neutral molecule. And that carbon is helping to create this organic molecule. It's a carbon-based molecule, neutral charge, and this is why it is so rapidly absorbed within that first hour or two, for example. So I think urea, it's a very different beast when we talk about it in the soil versus the foliar. And I'm a big, big fan of it when used through the foliar pathway. Here's a couple of quick examples of just the benefit of some of those carbon additions to nitrogen use efficiencies. This is a humic acid example. This is also from New Zealand. So some of you folks might have seen some of this work. But here we're looking at initially just comparing granular to dissolved urea, and you can see again dry matter production. This is per hectare initially, and you can see that there's a little nudge here. The liquid urea seems to be just kind of across 9 cuts on a pasture context here, seems to be outperforming the granular, the liquid versus granular there. But then if you look at the amount of production per unit of nitrogen, that's then where you see the big efficiency gains, because of course the foliar, you're applying a lot less on the foliar as compared to the soil. So when we then compare the efficiency of the dry matter production per unit of nitrogen, not per hectare, you can see that that's where we see these really big improvements. So not only have we achieved slightly more production, we've actually used a lot less nitrogen, thereby increasing the efficiency of nitrogen that we used. Okay, and here's just another example then, bringing in this carbon point, the humic acid, extra stabilizers, improving uptake, minimizing losses, that kind of thing. And again, you can see where we have urea added with a bit of humic, that extra carbon. You can see we see there slightly improvements in production, biomass production per hectare. And here we're looking at then a dissolved urea compared to dissolved urea plus humic acid or humic compounds. And you can see then the benefit of comparing just foliars now, one straight urea, one with the extra carbon. You can see that the blue bars here, we have this slight consistent increases in silage cuts here by adding the extra carbon stabilizer. So they do help. Okay, I'll lose my last 3 minutes. I'm going to squeeze extra to talk about then nitrogen use efficiency, another definition, because we always talk about nitrogen use efficiency in the general definition, which is really talking about the nitrogen uptake efficiency. And that's really defined as the total nitrogen in the plant relative to how much fertilizer we applied. So of the amount of fertilizer I applied, how much did I get into the plant? And sure, that's very important. We want to maximize the uptake efficiency to minimize the losses. But we also have another definition, which is the nitrogen utilization efficiency. And this is defined as the biomass production or the seed yield, depending on your context here. So the biomass production relative to the total nitrogen in the plant, reflecting the plant's ability to convert acquired nitrogen into biomass. So here what I'm talking about is, once we get nitrogen into the plant, how much biomass can I produce per unit of nitrogen? And to answer that question, we have to tease apart the different forms of nitrogen, because different forms of nitrogen are more or less expensive for the plant, more or less costly for the plant to utilize and to turn into protein. And the inorganic forms are the least efficient forms, and the organic forms are the more efficient forms, because the organic forms drain less energy for the plant to make use of them. And I'm going to summarize this really quickly so that we can wrap up and hit some questions and see what you all want to talk about from there. But here's a study that was really just kind of explaining this. The carbon bonus of the organic forms of nitrogen is what really helps them have an efficiency gain to improve that utilization efficiency. So the chemical form taken up, whether inorganic, like nitrate, or organic, such as amino acids, can influence the shoot and root growth and therefore nitrogen use efficiency. And the key point here is that the carbon cost of making use of organic nitrogen, turning those, like amino acids, turning those into proteins, that the carbon cost or energetic cost of that is lower than that of the inorganic forms. And it is the carbon bonus in those organic forms that ultimately make it more beneficial for plants to take up organic rather than inorganic. And this very busy slide is just illustrating this. When the plants take up nitrate or ammonium, there's no carbon there. And the plant doesn't want nitrate or ammonium. It wants organic nitrogen, like amino acids. So it has to take photosynthetic carbon and drain photosynthetic carbon to stitch together with that ammonium or with that nitrate to turn it into an organic molecule. So that's where inorganic forms are less efficient, because they are after photosynthesis, and we get now gaseous carbon dioxide into the plant and chemical energy, glucose. Now we have these carbon molecules. This is the carbon backbone that the plant has to now steal from or utilize to stitch nitrogen into to build all these various different amino acids that you can kind of see here. So point being, this photosynthetic pathway here, we are draining carbon. If the plant just takes up amino acids like these, if it's just absorbing amino acids, they've already got carbon attached to them. So therefore, we don't need to drain down on photosynthetic carbon. So we save photosynthetic carbon when we feed the plant more organic forms of nitrogen. And that photosynthetic carbon can be used elsewhere for greater biomass production, for more root growth, for more tillers, for more seeds, whatever. When you save photosynthetic carbon, it can be used elsewhere for biomass production. And that's why then the organic forms are more efficient, because they drain less photosynthetic carbon and the plant can use that elsewhere. And that's that subtlety about the utilization efficiency. And so I'll just whiz through this very quickly to say that nitrogen is not an island. It works with other nutrients that help it do what it does. And so when the plant takes up nitrates, it needs molybdenum, it needs sulfur, it needs iron to convert that nitrates along the pathway into ammonium. Once we have ammonium, the plant then needs manganese or magnesium to also carry that on into the amino acid, into the organic form. And then we have all of these other nutrients that are also very important for final protein synthesis. And urea sits back here too. Urea is a special example here. It needs nickel. Nickel is essential for the plant to convert urea along this pathway. And just to emphasize this point, the plant doesn't really want nitrates, doesn't want urea, doesn't want ammonium. It wants organic molecules up here. It wants amino acids and especially proteins. So if you have, for example, a molybdenum deficiency, you will have nitrate accumulating. You'll have a backlog. If you have an imbalance of any of these nutrients in the red boxes, this will create a bottleneck in which the nutrients will not move along this pathway optimized. So molybdenum deficiencies can lead to excess nitrates. A nickel deficiency can lead to an excess of urea. Urea is stuck as urea, and that can cause urea scorch. Urea scorch can, in some instances, not always, but it can be a nickel deficiency, and therefore urea is stuck as urea. So I'm just emphasizing here that nitrogen is not an island. If you want to optimize not only nitrogen uptake efficiency, but also the nitrogen utilization efficiency, you've also got to optimize some of these other important nutrients and trace elements. And that's where things like your plant analysis, tissue analysis, sap analysis, these types of things become very important to check the other minerals and make sure you've got the other synergistic nutrients to work with nitrogen. I'll skip over that one because I was just going to talk about some of those organic forms there being more complex, but more efficient. And yes, the plant can take up these larger, more organic molecules, and even as whole bacteria, as I'm sure many of you come across things like the rhizophagy cycle, even very large things like bacteria can even be taken up by the plant and used for nutrient supply. Okay, so in closing, foliar nitrogen, yes, it's a very important nutrient, important for production, we know this, but it's also very leaky. So we have to be careful with it. Judicious use is essential. We want to be careful. We want to be timely. We want to try and spoon feed. We want to be very targeted with our placement, with our application, with our rates in order to minimize some of those losses. There are many benefits to the foliar applications. We touched on some of them, like the efficiency gains, minimizing losses, minimizing lockups. You can choose the form of nitrogen, improving the efficiency through organic forms, as we kind of closed on there. Improvements in economics, you can use less, many benefits, but I emphasize it is just one tool. It's a tool in the toolbox, but in order to optimize the outcome of that foliar spray, the benefit of that foliar spray, you've got to think of some of those variables like the formulation, the application, the crop or the species, and those environment conditions. And I would emphasize, I think that the biggest gain, the low-hanging fruit here is just tweaks on the formulation and just getting that applied in the right environmental conditions. I think those are your low-hanging fruit. Tweaking the formulation with some of the things like water quality, spray pH, getting that out when there's high humid conditions, I think this goes a long way. We talked a bit about water quality, spray pH, including those carbon sources. They're good for chelating, for stabilizing, but also for improving uptake. And then some of those carbon sources are also carbon. So they're also saving photosynthetic carbon in the same way that we talked about there at the end. So the idea of kind of combining some organic forms like amino acids, like fish hydrolysates, with your synthetic forms like urea or UAN or whatever you're using, there is benefit to include some amino acids, some organic forms with those inorganic forms, because there is a synergy there between the two to optimize both uptake and utilization. And that closing point that I made there, just use tissue tests, use sap analysis, whatever you use, but make use of those tools, because it is important to check what other nutrients are limiting. It's not all about nitrogen. Yes, nitrogen is very important, but you've got to optimize the other nutrients that impact the efficiency of the conversion or the metabolism, the utilization of that nitrogen. And you can pile all the nitrogen on you want, be that through a foliar or be that through granule. If you have a molybdenum deficiency or a sulfur deficiency, that nitrogen is not going to be optimized being turned into protein. So you've got to step back, widen the lens, think about plant nutrition as a whole and where nitrogen sits within that. and yes, then work a bit more holistically with the plant nutrition there to optimize some of those other essential and beneficial nutrients that also improve the efficiency of your nitrogen utilization. And I mentioned I ran that foliar nitrogen course. Everything was recorded. It's online. If you want more information, you'll find that on my website there. What you saw today was a summary of a very deep dive into that topic. It was 10 lectures, over 10 hours of material, going deep into some of the things that you've heard the highlights of today. So you'll find more information on that on my website if you're interested. Okay, thank you very much. Sorry, folks, I did go a bit over time, but hopefully we've got some questions and we can get through them all, and I can stick around for a little bit if we need to. So I'll hand it over to you, folks. Thanks very much.
Awesome. Thanks, Joel. Really appreciate embodying that for us today. We've got a few questions that have come up from our members. We're going to quietly work our way through. One question that came sort of first, we'll start with Tony Joseph. Can plants take amino acids through their leaves, and what is the conversion efficiency?
Yeah, so as I kind of got there at the end of that lecture, absolutely. Amino acids are actually the fastest to be taken up through the leaf. They are the number one, followed by urea, followed by ammonium, followed by nitrate. So in that order is your speed of uptake: aminos, urea, ammonium, nitrate. And so, yes, they can be taken up. And that's, again, the reason they're taken up so quickly is that charge neutrality. And the benefit of that amino acid, once in the plant, is that it is already amino acid. It is already an organic form. So it's very quick and very efficient for it to then be converted onwards into those proteins and those other valuable organic molecules. So, yes, the challenge with amino acids is they are more expensive. The cost per unit of nitrogen is more costly. They save the plant energy. They're efficient in terms of saving the plant's photosynthetic energy, but economically they do cost more per unit of nitrogen. But that is offset by the fact that they are such an efficient form and more easily utilized.
Yeah, one of the questions, Joel, that come up when you're talking about water quality in the dairy space that we work in quite a lot. A lot of our clients will use the dairy effluent or dirty water as the base liquid to mix their fertilizer in. The question is, you talked about the clean water. Is there an issue using effluent with the likes of urea?
Yeah. Look, I know a lot of folks do that, and look, don't get me wrong, it's okay, but I'll be honest and say it's not my preferred for the reasons that I outlined. Look, some of the things in the dirty water is valuable. So some of the soluble nutrients out of the manure, some of those soluble carbon compounds, that part of it I would say is very beneficial. But in terms of some of the other cleaning chemicals and things that may also be in there, some of these may also then start to negatively impact. So look, I know on a practical level we've got to kind of meet in the middle here somewhere. And look, I know it's an easily available resource for you, and yes, I agree it should be put to use. So I support its use in a general sense. But if it's working for you, fine. But my honest opinion is, if you really, really want to optimize the foliar spray, you should really try and use as cleaner water as possible.
I'll go to the next one. I've got a question here from Rudy saying, as far as carbon-based inputs, what's the best sort of carbon that can be used, and should it be acidic carbon to reduce pH if possible?
Hi, Rudy. Yeah, look, I put that carbon slide, the carbon sources slide up there with a couple of different options. And my view on this is that I actually like to use a bit of diversity of different carbon sources. I think they do slightly different things. Some are better chelators in different contexts. Some will have stronger or less affinities for different nutrients and that kind of thing. So my general rule is I like to use at least maybe 2 different sources of carbon, 2 or 3 as an example. And because I wouldn't really say there is necessarily a best one. I think they do different things. So the amino acids, look, they are very good because they're small. They're very good chelators, as we talked about. And because they're very good chelators and they're very small, they're very quick to be taken up. So they are an organic source of nitrogen. So look, I am very partial to those because they tick a couple of boxes there. But the humics and fulvics, those have a much bigger exchange capacity, so they can actually hold on to a lot more nutrients than amino acids can. So you've got a much bigger sponge to complex and bind. And humics and fulvics, they're both biostimulants. Both of those have been shown to stimulate root growth through more biostimulatory kind of pathways. So they bring that added benefit of saying, well, okay, here's some humic or fulvic to stabilize the nutrients, but these are also biostimulants that can stimulate, for example, root growth. So so I think that you've got different kind of strengths and weaknesses of some of those different sources, and so I would generally use a bit of a mixture. And to your point about pH, just emphasizing that the one you have to be a bit careful of is the humic acid. It can be a bit fussy about being acidified. So if you are going to acidify the spray mix, be a bit careful with humic acid. You probably want to lean more towards the fulvic acid instead.
That's cool. We've got a question here around, you mentioned nickel being of use in that nitrogen conversion factor. What are you targeting in terms of the amount through either herbage in a pasture context, or an amount that you're looking for in soil, so that we've got something to kind of baseline to?
Yeah, good question. And I mentioned nickel with a bit of a warning. Of course, it is a heavy metal, and we do need to be careful of it too. It is also an essential nutrient, so we need some. I would emphasize a couple of things here. If you have very acidic soils, then that's going to make the nickel a lot more available. So I think I would only be a bit worried if we were talking more alkaline soils. I think if you're acidic soil, you're probably going to have enough in there. Such small amounts are required. And the other thing is that, particularly in a dairy context, if you've got manure and organic amendments and things being returned back into the system, you're also going to have some nickel in there. So my rule of thumb with nickel is that I would only consider applying it if it was an alkaline soil with no history of organic amendments. So I think beyond that, it's not too critical. And to answer your question, it's very hard for us to know exactly what the target range is in the herbage. We don't have that answer. The best answer I could give you is that it's somewhere similar to molybdenum. It's probably a little less than molybdenum, but somewhere around there. It's required generally for most plants. Maybe the exception would be some of your molybdenum accumulators like legumes and brassicas. But generally, it's required in similar amounts to molybdenum. But I would caution on applying it. Go slowly, just try a small amount first, be careful with it. You can use things like nickel sulfate, and we're talking something like around about 20 grams per hectare. It's very, very small amounts that you might kind of try.
Thank you.
Yeah, question here from Foster Joel. So in the grazing sense, obviously they want to minimize the window. The rotation is as short as possible. And as far as applying foliar after grazing, is there an optimum sort of cover that's required? And what do you sort of experience there?
Yeah, good question, of course. Yeah. And after your grazing event, you need a bit of time for that leaf to regrow. And so it's going to depend, of course, on the season. If it's very dry, the regrowth is going to be a bit slower. Under better conditions, it'll be a bit quicker. So it's going to depend on the time of the year and a few factors there, of course. But as a general rule, I think, waiting something like around about 7 odd days just for a bit of leaf growth and recovery. I know some folks that go a little bit earlier even. I think that's okay. But I think that you want to wait maybe at least a week or so for a bit of leaf regrowth to then have enough leaf area index to actually capture that foliar spray and make use of it. And I think that pastures are a very interesting model crop, so to speak. I think they are particularly favorable for the foliar approach because, we have a permanent perennial cover. We have a lot of leaf area. It's not a row crop where we've got bare soil in between. I think you've got a lot of leaf area, a lot of soil coverage. So I think they are a perfect crop to really work with this foliar approach. I think it's very, very beneficial, particularly in a pasture context.
We have another question here from Regan Bailey. Do you have insight into foliar P or phosphate uptake efficiency?
Yeah, a little here. I shared a study or two in the online course, particularly about FOS as well. So FOS, it can be taken up through the leaf. It is a lot trickier than some of the other nutrients, and it seems to be it does work and it can work well, but it does seem to be quite linked to plant phosphorus status, meaning if the plant is actually not deficient already in phosphorus, it doesn't seem to take up phosphorus very well through the foliar at all. But if you do have a phosphorus deficient plant, the leaf P status is low, then yes, foliar P can be effective. And there's also the discussion of spray pH seems to be a big one. It seems to be that quite acidified conditions, the spray mix is favorable for phosphorus. There was one study that I saw that suggested even as low as pH 2 was better for FOS uptake, and I'm not sure I probably would go that low, but in the study in the control conditions, they showed that at pH 2 the FOS was most effectively absorbed. So yes, it's possible, but I think plant P status is key, and definitely you want to kind of acidify your spray mix if you're doing FOS. Yeah.
I've got one here from Maddie. She's asked, is calcium through a foliar program likely to change the soil structure in heavy soils with high magnesium? And in brackets here, she's got 1-to-1 calcium to magnesium ratios. And would fine particle calcium hydroxide be suitable as a carbon source plus boron?
Okay. In terms of improving calcium status in soils and soil structure, I would say that improving soil structure, there's kind of two things here. One, it was the classic view of say, okay, let's apply some lime to the soil. Let's get that calcium into the soil. Let's flocculate that soil, open up that structure. And look, that is going to work a lot quicker than trying to do a foliar calcium and trying to kind of change soil calcium through that. It's going to be a very long and slow process trying to change soil calcium. They're not really quite the same thing. However, where your idea is interesting and a little bit on track here is that I would say there's two ways to restructure soils. One is with, yes, flocculation, calcium applied directly to the soil, but the other one is using plant roots and encouraging deeper roots and roots to penetrate down through soils and do more of a biological restructuring through root growth and death and root turnover, creating those channels and tunnels for successive generations of root growth to kind of keep regrowing and restructuring. Now, in that sense, that's where calcium as a plant nutrient, not as a soil flocculator, but as a nutrient, is very important. And that's where it could be foliar calciums could play a small role here, but is that when those roots are penetrating down into the soil, all of that growth is happening in the root tips, and the growth in those root tips is being driven through cell division, and it's calcium that drives cell division. So I would say that as an essential nutrient, yes, calcium is very important because of its role in encouraging cell division. That's going to help the roots push through into the soils and help with that biological restructuring. But I wouldn't say no. I would say that no, a foliar application is not really going to change soil calcium status. I think it would be a very, very minor, if anything at all, maybe in the long term. And the other part of the question is, yes, micronized type calciums and that kind of a thing can also be used. Boron, of course, is a calcium synergist, so putting boron with calcium is generally a good idea. They do work together. Boron is also, again, coupled with calcium in terms of importance in the plant, in the root tips and the shoot tips, all of that kind of thing. So yes, I would say that boron and calcium is a good idea to mix those together. I wouldn't say that the carbon component there is going to be enough on those own. I would be adding extra carbon additives. I would be adding some extra molasses, humics, fulvics, that type of thing to get some carbon stabilization of both the boron, especially the boron, but also the calcium as well.
That's
Awesome.
So you touched on some elements potentially being locked up in the soil, Joel. So I've got a question here from Brad. So he's just had a soil test done and had total P levels included. And The test has shown that he's well above recommendation. So does that mean the P is locked up? And if so, I guess what's some of the things that he could do to try and unlock that?
Yeah, that's right. So this is what we touched on a little bit in the lecture, and this phosphorus is a classic example of this, whereby many soils do have luxury reserves of many nutrients. They're all there, they're just often locked up. And some are particularly prone to this, and phosphorus is probably one of the best examples. And so if your total pool of phosphorus is adequate, you might take a standard soil test, which is only going to measure the more available fractions. And that soil test might tell you that the available fraction of phosphorus is low, is deficient. And therefore, the recommendation might be to apply more phosphorus. But actually, once you peer into the total pool, measure how much is actually there, but okay, maybe locked up, etc., but it is there, that obviously guides you to say, well, what's the point of putting more fertilizer on? I've already got plenty in the total pool. I just need to work on unlocking that. And that's where then a whole range of strategies become important. The longer term ones are generally trying to improve your soil health, more plant species diversity, more diversity of root exudates. It's going to drive a greater diversity of organisms in the soil to then solubilize some of that phosphorus. So phosphorus is very much a biological process in which it's solubilized from the soil. So I would say that optimizing plant species diversity, different roots, root depths, root architectures, all that's going to help to drive soil biology and drive some of that unlocking. And I shared the examples at the beginning of the lecture today where a foliar spray can actually stimulate uptake of soil reserves. And that would also be part of it, to optimize the plant foliar status in order to encourage those bigger roots and to encourage better root exudate production, organic acid production, which is then going to solubilize some of that phosphorus. So, so I would say that I would be using things like multi-species, but also optimized plant nutrition, and that's going to help to liberate some of the phosphorus in the reserves.
That's awesome. Joel, we have a really common thing called gibberellic acid that we use here in New Zealand, particularly on the shoulders of the season. It's a practice that's pretty broad and common. Can you comment as to your opinion on it, plus when it is linked with nitrogen, how is that a good thing, is that a bad thing, and what complications are there around efficiencies?
I've come across, I'll be honest, I don't know, I don't have a deep insight into this with a really good answer for you, apart from saying that I have come across it before in a dairy context. And I think it's, you folks in New Zealand that I've come across this a few times in Ireland, some people doing it. And I think they learnt it from you folks. I don't know deep about it, but I have heard that it's a good practice. And it's really, the point there is that you've got the hormone there to also then stimulate some of that root growth and some of that root activity. And yeah, look, it makes good sense to me. It's really the same point that I kind of made with those carbon sources, saying, well, look, let's use some of the carbon sources that can also stabilize, but also be biostimulants in their own right. And I think that that's where the idea of combining some plant growth hormones, like your example, with nitrogen or with foliar sprays generally, yeah, I support the practice. I think it's a good idea. But probably you guys would have a bit better local knowledge to comment on that. I don't have a deep insight to it, apart from I've heard of it. I've heard people are happy with it and it works well. And I think that as a general rule, yes, I like the idea of including essential nutrients and biostimulant type things together.
That's cool. Tony, I'll flesh it out a little bit. Super common. We have seen dry matter responses increase in pasture, but it's because gibberellic acid will elongate stem cells. Personally, I'm not a big fan of it, just because once it's applied, it seems to have a phase where it will pull growth forward, but then it goes through quite a big recovery phase or process after. And so if we think about when we use it in a dairy type context, we're trying to shift weight forward, but then we really gun for that, try and get cows to peak, but then it shows up as we're kind of like getting through that peak milk, heading into mating, we just go into a real slammed hole right over when a plant wants to go to seed. So if people do use it, I sort of caution. Personally, I sort of, we've noted it drops bricks and a few other impacts that can be a little bit more confusing. However, it does grow. So it's a tool that has a place. The goal would be to not get into the position that you're tight for feed and you're having to pull on it, but it does have something to offer. It just kind of has to be managed well in the bigger sort of picture.
Awesome.
Got a question, Joel, here from our partner Alejandro in Uruguay. He's asked, what's the differences between a sap analysis and a tissue and leaf analysis, if any?
Good question. We could talk on this one for another hour, probably too. Look, I'm a fan of both. You know, the traditional analysis is the standard tissue analysis where you're going to sample the whole leaf and dry down that whole leaf and digest the whole leaf, versus a sap analysis, which is really more of a leaf extract. You're kind of, the analogy would be it's kind of like a blood analysis for a plant. You're just extracting an extract and trying to assess what's in the sap. And they give you slightly different things. You know, one is a bit more longer term supply. One is a bit more of an indicator of a snapshot of right now and what's kind of in the plant sap and that kind of thing. They are different. They've got different kind of ideal levels. I'm a fan of both. I know that the sap analysis is becoming very popular in more recent times, and I think that there are a lot of good things about it. Particularly the sap analysis, I think, is very good. The way you know, you can often sample the young and old leaves and get a bit insight into the nutrient mobility, but also on that sap analysis, you can kind of really get a picture of both the total nitrogen as well as the nitrate and ammonium fractions. And so that really helps you to tease out what is the balance between those inorganic forms and the beneficial organic forms. So I think there's a lot of strengths to that too. But you know, the traditional tissue analysis, it has a lot of long history. We have very robust kind of databases of what the target levels and what the ideal levels should be. The sap analysis is still emerging. We're still fine tuning exactly what kind of some of those ideal levels should be. So look, I think people are using both. There's a bit of different context around them. I think both have their place. I rather than stress too much about which one's better or worse per se, I think it would be more important just to make sure that you're using at least one of them and doing that kind of regularly and tracking the trends and patterns and things over time.
Awesome. We had a question come in around how much water per hectare is roughly required for effective foliar application using your tone Fert units. Might be more of a question for you, Tim.
Yeah, I guess I can speak from just our experience here. Obviously, the machine, basically, you can really tailor it to exactly how much water you want per hectare based on environmental conditions, etc. Typically, we're seeing things between 100 to 150 litres a hectare is common, but it can be tailored based on a lot of things, really, based on rate per hectare of your exact product and time of the year, etc.
As a general rule, I usually work off around about 100 litres as well. I think that's a good amount. And, you know, I hear both sides of this. You know, obviously it depends on the size and scale. Larger farmers are often trying to use less water per hectare. So, you know, it's not uncommon for some folks to be going down to 80 or 60 even, you know, maybe in more of a cropping context. But I do know folks that are also going higher, 250 or 200, and they find that extra water rate also, it just helps to add to kind of increase the humidity in the canopy, in that kind of micro humidity around the plant. So some people go for a bit higher water intentionally just to try and get the humidity up in the canopy. And again, as we talked about, that can improve the uptake as well. So I think that makes a lot of good sense too.
Joel, we had one quick question. I've just answered on a chat, but just can you talk a little bit about cobalt and importance around legumes fixing in? It's a topic that probably as New Zealand we're really growing in our understanding of, and it's kind of gone a little bit under the radar for a long time, and many people have really good clover levels, or we at least plant it, but then we're not really fixing nitrogen that well. Can you kind of just touch on that a little bit?
Sure, yeah. You know, there's also quite a few key nutrients that are required for legumes and for the rhizobium to do that function. And cobalt is one of those, along with others. You know, molybdenum is also a really important one. Molybdenum and iron. Nickel also plays a bit of a role in ant fixation. Phosphorus for energy. For legumes particularly, we could add things like calcium and cobalt, as you say, boron and copper. All of those nutrients actually play a role for either the bacteria and or the plant to support those processes. And yeah, you're right, cobalt is one that's particularly important specifically for legumes. Of course, it's also part of the B12 for the nutritional value of the pasture as well. And so yeah, there's been many good responses for plants, and that's that kind of thing where on legumes, it is very important to kind of dig up those legumes and have a look at those nodules. Often the legumes can be growing well and healthy and functioning. Sometimes they can even have nodules, but they're not necessarily functioning nodules, not that nice pink color inside. So yeah, I've seen some incredible responses on legumes from, for example, a foliar spray of molybdenum that was deficient it was in a bean crop, which then just absolutely exploded with nodules after that foliar application. So look, I would say that list of nutrients that I mentioned there, they're all kind of important. Cobalt is on that list, and if cobalt is a bit of a common limitation or something for some of your soils, or is something that has maybe been neglected a bit in recent decades, I think that it is a trace mineral that is required also in quite small amounts, that is very appropriate, most effectively addressed through a foliar spray. And I think, yeah, the plants can make use of that, send that down to those nodules where it can support the end fixation process.
Great.
Another question we get commonly asked, Joel. Yeah. We've got a question here from Foster, and I'll elaborate on it. He's saying, do we need to apply foliar in every grazing, or can we build levels in the plant with one higher rate to cover the plant's N requirements over several grazings? And I guess some of the feedback we get from people that I speak to, they say, yes, it works quickly, but it doesn't run out compared to the likes of a granular application.
Yeah.
Can you speak to that?
Yeah, and as a general rule, I would say I agree with that. I think that is as beneficial as foliars are. I'm a big fan, as you can all tell. We also have to be honest about, like every tool, it has its pros and cons, strengths and weaknesses. And I would say that is one of them, which is that, the max amount of nutrient or nitrogen, in this example, per application, is a lot lower in a foliar as compared to a soil. And that's good and bad. That opens the door to saying, well, okay, therefore we need to kind of do this low dose spoon feeding approach, more frequently low dose. And that's part of where the efficiency gain comes in, is because we're using less, we are synchronizing supply and demand from the plant. It's when we apply granule at a higher demand, at higher supply, that yet the demand is not there. That's where we open the door to all those losses. And yes, it's fair to say that, sure, some of that nitrogen can be held on the soil, and it is, and therefore a granular can give you a more sustained response. I think that is a fair comment and a fair critique of that would be a weakness of a foliar spray, is that, yes, it does run out of steam a little earlier than what a granular would. But I don't know, I don't totally necessarily see that as a problem. I think that it's okay to be doing more frequent spoon feeding. That is where some of the efficiency gains can be had.
So one more question here. Joel, there's a common question that's coming up in thread, and I'll try and refabricate it. Peter has brought it up, and there's a couple other people, a guy called Ty Cohen. When we're using either stickers, oils, or we're looking for amino acids, do you have a way for us to digest, like, you look at a product and then we say, okay, it costs X, and then we have a look at another product and it costs X. For us as farmers, it's so hard to know, like, yeah, that's a great option, or actually, no, that's not a good option and this is good. Do you have some kind of rules of thumb that you run by around how we should kind of see it, or how we should be able to digest it to kind of make a start somewhere if that's new for us?
Yeah, okay. That is a tricky question. I agree, and I sympathize with you on this question, because there's so many products and things coming out on the market these days as well. It's becoming a very saturated space, and it is very hard to kind of separate, the chalk and the cheese kind of thing. So I don't have a great answer for you, I'll be honest, apart from saying that I would start with taking a recommendation from another farmer that I know or trusted, get some opinions out there of some other farmers and what they're using and whether they're happy with the product. So I would start with kind of taking a recommendation. And I think that then, of course, it becomes a little bit of maybe just getting a small amount in and doing a small trial to start with. You'll have your standard approach. Get in, if you're going to try a new product, get in enough just to try it on a field, or split a couple of fields in half and do half with your standard approach. And just a small amount there. I know that I don't expect you to be going to run a triple replicated study per se, but even just a simple kind of, okay, I'm just going to compare it to what I'm doing, split a field, do a few observations when you're back in that field, and just kind of experiment to compare with what your standard is. And I think that on some of the, I have a bit of a better answer on some of the other biostimulant type carbon additives, maybe not the stickers and things, but maybe some of those humics or fulvics. There's lots of these different types of things out there too. I would say, I think one of the kind of good strategies to try and kind of compare different products is to do a little basic kind of simple seed germination test. So even, just in a little Petri dish, do a little seed treatment and look at the effects of that humic or fulvic or amino or that type of thing on kind of germination, on sprouting. It's just a quick kind of 7-10 day, 2-week little experiment where I know that we're asking two different things here. One is like, well, what are the effects on germination versus what are the effects on a foliar additive to improve uptake? I acknowledge these are different things, but it does, the simple seed germination test, it does give you a bit of an indication of the general biological activity of the compound. And if it's got good effects on general biological cells and on that seed germination process, it likely follows that it would have beneficial effects on leaf cells as well equally. So that would be something I would use to maybe compare a few different products in a simple way.
Great, thank you.
I think that about covers our Q&As. It's been amazing, Joel. We probably could have had you for another 10 hours if we needed it. I've definitely got my head full of ideas now. And I'm not a farmer, but I have been following your course, and I love it. So anyone here that's listening and does want to learn more, definitely go check out Joel's foliar nitrogen course. It's a great resource, and you can do it in your own time, which is great. And yeah, the visualizations that you showed, especially around the nitrogen converting and things like that, it's just nice to see all these visualizations as we see them on farm, and we see the benefits our users are getting from their machines, and we see the results, but it's really nice to look at the why and really understand what goes on under the surface, and really puts emphasis on people reading their soil tests and talking to agronomists and actually getting that balance right so they are using their nitrogen efficiently. So yeah, thanks from the team here at Fert. And do you have anything to say there, Canaan?
No, thank you, Joel. It's really hard to elaborate for our kind of wider New Zealand audience, but Joel has been around the block for a while and is really powerful in what he communicates in this sector. So we don't actually have resources like this that we pull on very often here in New Zealand. So Joel, we really appreciate you coming in, buddy. It's awesome, and thank you, Fert, for making that happen.
Yeah, my pleasure, folks. Yeah, anytime. I hope we can collaborate again another time. And yeah, thanks for everyone's questions, and great to all have your input here today. And yeah, if anyone's interested, on my webpage, I do have a resources webpage as well, and there's a few articles and things that I've written about various topics. So you'll find a bit more information out there if anyone else is interested.
Awesome.
Great.
Cheers, guys. We'll be
Awesome.
putting this on YouTube. So if you came along halfway and want to see the start, just keep an eye over the next couple of days on our YouTube channel. You might see it pop up on Spotify as well. So there'd be a chance to go back through and listen to it all if you weren't taking notes. But yeah, so thanks, Joel. Thanks everyone for coming, and we'll see you guys on the next one.